Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome everyone to the Genetics Podcast. I'm very excited to be here for a super special episode for me, actually, with Dr. Anthony Bleyer, who's professor of medicine at the section of nephrology at Wake Forest University and adjunct at Charles University in Prague, and his close collaborator, Stan Kmoch, who's leads the National Infrastructure for Medical Genomics in the Czech Republic. This episode is special for me because I've mentioned a few times on this podcast how we have a genetic kidney disease in my family, and these are actually the two people I think most responsible for cracking the code of what's going on in our family, but also a number of others, and really hundreds of others over the last few decades as they've worked across some of these mysteries and inherited genetic kidney disease. So we're going to have a little bit of a conversation about their work more broadly, but I'm really excited to do this because it's very rare that you get to have this kind of journey as a family with a rare disease, that someone spends years and decades to actually crack the code and figure out what's going on. So it's very cool to have this conversation, have a quite a personal connection to it. So Stan and Dr. Bleyer, thanks so much for being with us today.
Thank you very much.
I'd love to start actually with how you all got connected from Wake Forest, North Carolina to Prague in the Czech Republic. You don't always get collaborations that span that kind of distance. Is there an interesting story there, or what brought you two together to work on these kinds of projects?
So I would say kind of goes back a little ways. So I'll start that I'm a clinical nephrologist. I did my training at Hopkins and the University of Pennsylvania. And I came down to Winston-Salem, North Carolina, at Wake Forest, and I thought I would be an epidemiologist. And in 1996, a family doctor named Arch Woodard referred a family to me with inherited kidney disease. And it was a disease I had never heard of before. The family had gout and kidney disease. So I went to the mountains of North Carolina and we collected some samples. And being a nephrologist, I thought I would check the urine samples. And so I went to the lab and that did biochemical analyses of the urine. And Dr. Suzanne Hart told me, why don't you start working with my husband to find this gene? And I said, well, I thought your husband, Tom, was a periodontist. And she said, he is a periodontist, but for genetics, you just have to tell him who's affected and who's not affected. So we started working on this project to find this gene. And in 2002, Tom was able to identify a mutation in the UMOD gene, which encodes uromodulin. And uromodulin is the most common protein that's excreted in the urine. And so we started working on to understand this disease. It's dominant negative, and the mutant protein is deposited in the thick ascending limb cells. So at this point, I thought, well, let me just see how many families I can find. So we started a web page, and I became what I didn't realize then, but what I call now is a rare disease clinical champion. In that I think like there's so many rare diseases and doctors may see one case in their lifetime.
Right.
And so it's good to have like one person, a clinical person or several, and who can really be a resource for families and help to direct them and hopefully help guide research. So we started getting referrals off of the internet. About 1/3 of our referrals come directly from patients. Surprisingly, their doctors, haven't reached out to us, and the patients reach out like you guys did. And
Yes.
so we started working, and we were able to identify the UMOD gene, but then there were a group of families, and we didn't know what they had. And by a stroke of good fortune, one of the families worked with Dr. Eric Lander at the Broad Institute. And so he started to help us, or we started to help him, I should say, to find a mutation in another gene called mucin 1. And so this gene, Stan will be able to give more details, but the mutation lies in a variable number of tandem repeats region of the MUC1 gene, and it's was very difficult to find. And when it was found, we could not do just Sanger sequencing to identify the mutation. So we're able to identify a lot of families who had one particular mutation, a cytosine duplication, but the other families, we weren't able to determine if they had a MUC1 mutation. Well, my colleague, Dr. Hart, went to the NIH to the dental branch and had to stop the renal research. And so this was a big problem. So I was like Moses in the wilderness, in the desert, and I had no one. And so then we read an abstract by Stan's group, about a mutation they had found in renin, or pathogenic variant in renin. And we looked through our families, and sure enough, we found the mutation pathogenic variant in renin, and we contacted Stan. And this was really great for me because I really needed a scientist to help me. And I think Stan benefited too because we had a lot of cases and we could really do the clinical stuff. So he invited me to come to Prague, and I took my son Tony Jr. and we had a great time. And I don't know, many years later in multiple publications, and we've been working together. Um, there's a book called The World is Flat, which says that by Tom Friedman, that everything will be done where it can be done the best and the cheapest, and
Mm.
the internet and FedEx will sort out the rest. And so that's us. I Skype with Stan every day and we talk about everything. And then we just send the package every couple of months, every month actually. And he does a lot of the brute force genetics.
Right. And so Stan, you're doing genetics, you're also probably doing other kinds of omics. And I know your interests are not just in kidney disease, but you also work across metabolic disorders. I'd love if you could talk a little bit about your how you met Dr. Bleyer and the team, but also a little bit about your work more broadly to give people some context.
Okay, so I started personally 35 years ago working at the Institute for Inherited Metabolic Disorders, which was one of the first centers to screen for phenylketonuria, like nationwide. And we had a very great tradition in metabolic profiling. And I actually started with mass spectrometry profiling of urinary metabolites. But in early 90s, I went to Denmark to Aarhus, and I learned some basic molecular techniques there, like sequencing, PCR, cloning, and so on. And it was a team of Niels Gregersen, and his team was interested in defects of fatty acid oxidation. So I brought that subject back to Prague, and I started to work on this subject a little bit. And because the beta oxidation is also in peroxisomes, then I met the Ron Wanders, who is famous guy in peroxisomal research. Then I spent some time in his lab in Amsterdam. So I learned like basic techniques because in early 90s, the science was not as much developed as I would say in Amsterdam and Aarhus. And there was the other great opportunity I had. I met Wilhelm Ansurge. Wilhelm Ansurge was a guy who developed the gel sequencing technology that was later commercialized by Pharmacia. And thanks to Wilhelm, he led the group at the EMBL in Heidelberg. And thanks to his support, we get one of the first sequencers in Czech Republic, and we were also able to operate his prototypes of Arrakis and all these fluorescent labeled dideoxynucleotides for sequencing. And starting with this equipment, we were the first lab in the Czech Republic who provided the sequencing expertise to different subjects. And I started with metabolic disorders first because it was my original affiliation. And then we figure out that the sequencing is, I would say, a very efficient tool to other medical specialties. So we somehow start to build up the much bigger and bigger facilities, always equipped with the top art instrument. And this, of course, we had a need for bioinformaticians, and then we get a number of variants of unknown significance, which meant that we had to develop tools of molecular biology to characterize these variants. And then we started to be interested in some sort of treatment. So we came to the field of developing the model organisms and trying to find some treatment modalities. So now we are a team of about 50 people operating in Prague, and we really provide very comprehensive expertise from starting from genomic sequencing to animal models and finding the new molecules and so on. And one of the projects, and I would say that like the most extensive or extended, is a collaboration with Tony, because he's a great clinician. He's always tried to help patients and families, and we just provide. So we serve to his desire, and we provide what we can.
Yeah, amazing. I want Oh yeah, go ahead, Tony, please.
Isn't that great? I bet every clinical researcher wish they had a geneticist, genetics researcher who could just help them. It's just awesome. It's really been great.
Yeah, I mean, you made a good point in your story though, which is that it's hard, almost impossible to find the full complement of skills in any one person, right? A doctor doesn't always have the genetic skills. Geneticists, as one, don't have the clinical skills. So you really do need teamwork to figure these things out. But that means coordination and collaboration, which can be challenging. In inherited genetic kidney diseases more broadly, if you could break it down, Tony, there are a couple of the common causes that you mentioned, like UMOD and MUC1. But for people who aren't as familiar, if a family like ours comes and says, we have clearly have something going on in the family tree, but we've never been genetically tested. What proportion of people end up with a cause ascribed today in one of these more common genes, or one of the rarer ones that you and your colleagues have discovered? And then what percentage are still in the no molecular diagnosis bucket, just to help people orient on what the problem space looks like?
Yeah, so it's, nephrologists have typically been more physiologists than diagnosticians. And so, believe it or not, 25% of patients who actually enter dialysis, so the end stage kidney disease, do not have a diagnosis.
Wow.
And it's just incredible. And a lot of times, we haven't really been good at pursuing diagnoses. Another problem is that we don't learn very much about genetics in medical school. And, for all 4 years, maybe a week or two of genetics. So that's made finding the causes of inherited kidney disease difficult. We've known for a long time that there's polycystic kidney disease and Alport syndrome. Those are the most common causes. And then Black patients, colleagues, Dr. Martin Pollack and Dr. Barry Friedman here, have identified APOL1 as a cause of inherited kidney disease. And then there are a host of many other diseases, I think like many other systemic disorders, where you have the big 3 or 4, and then there are like 50 or 60 other causes of kidney disease. And so about 10%, in a study by Dr. Groupman and Garavi that was in the New England Journal, about 10% of patients with kidney disease at least have a genetic cause. And early on, when we started, it was really expensive to get any genetic testing done. And now genetic testing is much more available, and we have multi-gene panels. So I think those are becoming incredibly more used. But then the nephrologists don't understand how to interpret variants of uncertain significance. And we've had another problem with our inherited kidney disease, the MUC1 disease, is that it doesn't lend itself to a typical genetic sequencing. And so a lot of times people will get the genetic sequence done and they're like, didn't show anything. And the doctor's
Right.
like, well, that was 300 genes. I mean, we did everything we could.
Right.
So our job gets a little bit more complex. But yeah, those are the most common causes. And then there are a host of less common diseases, but they may be become more common as we do more testing.
Yeah, I maybe we could dive into the case that I'm very familiar with, which is the new gene that you discovered discovered the link to kidney disease, which is ApoA4. I'd love to hear the story here because you have five families. I think as of the publication, we'll link to the publication in the show notes for people who are interested in it. But I spent my PhD working on rare neurodevelopmental disorders and doing gene discovery. I know how hard it is to make these discoveries, and I remember when you sent the manuscript, immediately I looked at it and said, yeah, I think they've nailed it because you have this cluster of missense mutations in a very clear place. So I'd love to hear the story there because, we had been part of the program for probably a decade before you made the
Yes.
discovery. And as a researcher, I know that that's how long it takes to get to the scale that you need. But sometimes participants don't understand that, right? You get tested and then years go by and the researchers are working hard in the background, but you have this just scale and signal to noise problem that you have to overcome. So maybe you could both talk a little bit about that.
Yes, I'd like to talk a little first from the clinical end. Boy, you hit the nail on the head because we get these samples. And so one thing, so we've been referred 1,000 families. About 300 have UMOD mutations, 250 have MUC1 mutations. But then with families, other families like yours, we had two problems. One is we had to rule out MUC1 as a cause. And then but we could never be 100% certain we had ruled it out. And
Right.
then we had to look for other genes. From a clinical perspective, starting as an epidemiologist, the statistician would always say, I need more numbers. And that's one thing that doesn't change. Whenever we find a new family, Stan says we need more samples. So we were referred a family from New England back in, first of all, a family from Indiana in 2012. They looked like they had what we would have considered a MUC1 mutation, but they did not. We collected samples, but it wasn't a super large family. Then in 2015, we were referred a much larger family in New England. And that family, though, it took us a long time to get samples on. And you have to gain trust. And so I have great colleagues, Dr. Kendra Kidd, we have Lauren Martin and Abby Taylor in our lab, and they just worked with these families. And over time, over many years, we got samples. Your family you're right. I got an email from your dad in 2015, which you had written several paragraphs on. And so we started getting samples, but again, we were focusing on the larger family. And finally, I think we had enough samples that we were able to really do something. And I'll let Stan take over from that point as to what happened.
So what we did, just the routine. So we perform either whole exome or whole genome sequencing, and then we look for shared variants. And the strange thing here was that we end up with one missense mutation in ApoA4. And when we did the Sanger sequencing and genotyping to study the segregation, then we figure out that there are a couple of people at one stage who has the mutation. And there were also some other individuals who had the kidney disease and had no mutation. So it took us, and ApoA4 was not the eminent candidate to follow up like directly because it's produced in intestine. And we did know that there exists the situation of ApoA4 amyloidosis. And in these situations, we are like following this lead and we are trying to understand what the mutation might do. And here, the simplest thing was to stain the kidney biopsy for ApoA4 distribution. And that was exactly normal. So we really were somehow stuck there because the segregation was not complete. The ApoA4 staining in kidney was comparable to controls. And so we look for some other candidates. So we did the whole genome sequencing. We follow a couple of other leads until we get the deep kidney or I think it was an autopsy or
Yeah.
a kidney.
No, I
There
can.
was a
So yeah,
kidney removed,
yeah, let me
and
just, if I
yeah,
could just break in. So yeah, so what happened was it all just came together all of a sudden because what happened is if you biopsy the cortex of the, so what happens, the mutated ApoA4, so ApoA4 is synthesized in the intestinal wall, so not in the kidney. So
Hmm.
that threw things off a little bit
Right.
initially. And then as patients have older patients, older age of onset of kidney disease, so we may have some patients who have kidney disease from something else, and then some of the patients were mildly affected. And so that was all what Stan was talking about. But then the ApoA4 forms it's synthesized in relation to fat ingestion. And then the monomer is filtered by the glomerulus and reabsorbed in the tubules back. And
Yeah.
so what happens in this condition is a pathogenic ApoA4 deposits in the medulla of the kidney, middle of the kidney. And we do biopsies of the cortex of the kidney.
Right.
And so we had several biopsies, and like Stan said, they were all the cortex, and so they didn't show anything. And he still persevered though, and he figured out it was ApoA4. And just the week that he called me about that, one of our patients was having a nephrectomy. And so I
Wow.
actually went up and it was in Boston, and I reviewed the slides at the microscope with a very renowned renal pathologist, Dr. Helmut Rentke, and he saw the amyloid in the
Wow.
medulla, and we sent it to our friend Dr. Nelson Lung and his colleagues at the Mayo Clinic, and they did mass spec, and they were like, "Hey, this is ApoA4," and so it was really incredible.
Yeah,
Yeah, it's
and
amazing.
yeah, interestingly enough, because they can also do the amino acid sequencing in amyloid, and they figure out that the ApoA4 that's deposited is the mutant form, which was
Oh, yeah.
like the last information we needed. And we also did some experiments in vitro where we are showing and we are observing now that if you mix these mutated ApoA4 proteins, they really stick together and made the amyloidic forms. An important thing here was that Tony is collecting these families over time. He's got over 1,000 families referred. Then we quickly just look into our exome or genome data, and we figure out that there are two more families with another variant in ApoA4 with exactly the same phenotype. And that was the final proof that this variance in N-terminus of ApoA4 leads to amyloidosis.
Yeah, it was really incredible because, our team, our motto is to help one patient, one family at a time. And we had families like yours that we had worked with since 2012. And so the first thing Stan did was look for the mutation that we had found in the family from New England. And there was also a family from Vermont that had the same variant. And then a funny thing was, we had gone to Canada many years ago, and after finding a gene for the Acadian variant of Fanconi syndrome, and we obtained a genetic sample on a patient from Canada. And that patient, which we had in our freezer, he had the same
Wow.
mutation. So the three families, so of course they all came from Canada down to New
Interesting.
England. And we worked with the Dr. Andrew Orr, who had many samples, and he sealed the deal. And then Stan said, "Hey, there are two other families." And we were like, "Oh, this is so great." Because we had been working with these families, your family and another family so long. And it's just so, it's the scientific part is really cool. But the other part that's really cool for me is I walk into our lab and I'm like, "Hey, we got it." And everyone's like, "Wow, this is great." So it was really cool how it all came together and how just having this big volume of families was really helped us to figure it out.
Yeah, if I could paraphrase the mechanism, and you all correct me if I'm, or the hypothesized mechanism, just to recap for everyone, what we think is happening is the ApoA4 is being produced in the intestine. the missense that you have two different missense cases that you've picked up in families, and something about the way the protein is folding means they aggregate, and as they make their way through into the kidney, they build up, and over time, effectively, you get the progressive breakdown of kidney function. And you can see that, you could see that when you then stained the medulla in the kidney and could see the buildup of the amyloid there, basically. And I could see how this was probably really puzzling at first because when you look at the gene expression of ApoA4, it's basically only in the intestine, right? So you're thinking I'm looking for a UMOD or a MUC1 or something that looks like it's this kidney protein, but then you have to put together this broader story. But I guess that leads you towards a potential therapeutic application, right? Can you block production of the missense protein in the intestine or prevent it from aggregating in the kidney in some way? I'm curious how you guys are thinking about that.
Yeah, so I just like to say just another really great thing that happened was when Stan told me ApoA4, I was like, man, it sounds so familiar. And then I thought, oh, that's someone's license plate in our parking garage. And it was Dr. Richard Weinberg at Wake Forest. He has a clinic next to me who studied ApoA4 for 30 years. So,
Right.
wow.
That's
So
amazing.
that was really great. And so he had actually done a lot of work on ApoA4. And one thing we know that ApoA4 is not really important in fat, absorption or in your lipid panel. It's not, doesn't affect your LDL or HDL very much. And so we're interested, potentially how a low-fat diet could affect production of this. We'd also like to be able to silence the mRNA or any other, I know you have a lot of listeners, Patrick. I'm sure some of them might have a
Yes.
great idea. Please contact us. We'd like to crowdsource this if possible. So, but Stan, what are your thoughts too on treatment?
Yeah, you already said it. So, reduce fat intake, maybe use some compounds that uh, block the reabsorption of fat in the kidney, and that's it. And then we have a couple of, I would say, the genetic
Yeah.
approaches, but they are far away from now.
Yeah, I guess, yeah, like you mentioned, RNA medicines or small molecule inhibitors or something like that, right? But I mean, it's interesting that the low-fat diet is a very attractive option for obvious reasons, right? If you can, and I remember when you shared this doing a little bit of reading at the time, it's specifically long-chain fatty acids, I think, that fire it up, right? So you could even get more specific into coconut oil is okay, butter is okay, but avoid peanut butter or those kinds of things, right? Yeah, I'm
No,
speculating a little bit here, but
yes, I agree. That's a theory that we're hoping we could do it. But the thing to me that was so heartwarming is when I work with Stan's team in Prague, they care just as much about the patient as we do. As I find with many, people I work with in industry, everybody really wants to help patients. And I've always, it's just great. We met with one of the families and they were just so happy to see Dr. Kmoch and hear it like firsthand how he found this. And so it's just, and the fact that his lab has all these areas that they can express the protein and do all sorts of things, that's just been an added bonus.
what are you all you kind of have closed the chapter, I think, on the discovery of this gene. I'd love to hear about some of the other things you're working on. You've got many other families in the program that you're working on new discoveries. What other kinds of things are you all focused on or working on together?
Yeah,
Uh,
as I told you, so Tone is referring all families he came across, and we just proceed through the standard process. We just sequence and we are trying to understand what the genome is telling us. So as in genomics, you always, for every family, for every individual, you identified something interesting, like the candidate variant. So
Yeah.
what we learned in the past is that we really look for the genes and mechanisms that may contribute or can work in a common metabolic or biological pathway, like endoplasmic reticulum stress, Golgi apparatus stress, and so on. So it gives us more and more sense some of the variants we are finding. But the problem is to prove it functionally, genetically, because as Tony told you, there are like three major genetic kidney diseases: PKD, Alport, and now UMOT and MAC1. And the others are like singletons, so it's difficult to and the families are pretty small. So one of the most, I think, interesting findings from the past, or from the recent past, is identification of mitochondrial DNA mutations that are causing the chronic late onset chronic kidney disease. So there are a couple of, I would say, very extended, because of maternal inheritance, of very extended pedigrees that show up the chronic kidney disease in their 60s or 70s. Right. So this is, I think, one of the hottest topics we are now following.
Interesting. Tony, how about from your perspective?
I would say, from my perspective, I've learned a lot about the diseases and how they affect families. And so we start out right with one or two families. We need two, at least, to make a genetic diagnosis or a new gene. Then from 2 to 10, we collect data and we kind of have an idea about the disease. Then when we go from 10 to 100 families, now we can really clinically characterize and come up with hypotheses and ideas. And along the way, we have to help the families. We have to help them find a diagnosis, find us, and then patients will ask, many questions relative to their disease or clinical condition. Will this affect me if I have a pregnancy? How will other things affect me? And so we try to work with the families, establish patient foundations, and just keep pushing the research. So that's a lot what I'm trying to do, is I'm trying to, with all these diseases, to create like this foundation, because it takes patience, both types of patients,
yeah,
right?
That's right.
It takes, scientific researchers like Stan, and it takes clinical researchers. It's like that three-legged stool, and we're all working together. And one of the things I'd say that's been the greatest in my career is just meeting these wonderful people, these wonderful patients who all help in their own way. Like the patient who told me, "Hey, I'm getting my kidney out."
Right.
What a great
Helps crack
help.
the
Or
code.
let
Yeah.
me give you a sample. Or and then scientists. And so that's been the best thing. But we're just keeping moving forward, trying to resolve more rare diseases, and we're really trying to find more ApoA4 families. And so we're really interested in that.
Yeah, are there are there international collaborations that allow you all to search in other kidney disease databases? Because yeah, I've got to imagine there's other programs. There's Genomic Medicine Service here in the UK. I'm curious how built out that is, where you can say, look what we found. Please go check your database. Obviously, people read the literature and they do that somewhat organically, but it would really help with the gene discovery process, wouldn't it, to have a master meta-analysis?
So I could comment, so Stan, what would you say? We use like Gene Matcher, right? To find, that helps us find new variants. And then I think one thing that's been really good is just our collaborations with many different rare disease researchers and getting the word out. But I wish we had a super huge registry. That would really be helpful. And we do look at various, obviously, genomic databases.
Yeah, Stan, do you have any perspective on that? Why isn't there a super huge registry? Any idea?
Yeah, I think in Europe, the genetic kidney research was, I would say, scattered. There are like groups in France, Germany, Belgium, Spain, that they felt like more the competitors than the collaborators. And we somehow ended up working with Tony. And then we figure out that there's a great physician and nephrologist in Ireland, Peter Conlon. So we started a great collaboration with Peter Conlon, and then with people from Cyprus. And year, there was the consortium, the European consortium formed, and it seems that things will change in near future, that people are trying to really come together, work
Yeah.
together, and at least exchange the information, for example, on new genes, collaborate and provide, exchange the materials. So I think the things changed a lot over past couple of years, at least in Europe. I don't know exactly the situation in US, but in Europe it's changing. There's Olivier de Woest in Zurich, and people in UK, Daniel Gale, and so the collaborations it's really coming up.
Tony, I wanted to ask you a question, which it comes up probably a lot when you do this sort of thing, and maybe it's related to, you made a really interesting point earlier, which is your field has been more focused on management than diagnosis in many cases, and one of the big questions in management is transplants and 3D printed organs and reprogrammed pig kidneys and those sorts of things. I understand that the pig kidneys are a lot closer to prime time than maybe they were 10 years ago when it was felt like a little bit more of a sci-fi thing. I'm curious to get your perspective on that because it is happening now, actually, right? There are some pig kidneys being transplanted into humans.
Yeah, Dr. Leonardo Riella, a friend of mine, he just published in the New England Journal their pig kidney. I'm super, super excited about the pig kidney because I look at like it's going to be really hard to make genetic changes in human kidneys.
Yeah.
But with the pig kidney, every generation we can modify the pig a little bit more. And I think I'm very much looking forward to that in the future. And I think that's one thing we're going to concentrate on. And I think that could be really great for our patients. Especially if we can make one that's HLA compatible and won't be rejected, we could talk no immunosuppression, and it might be better even to just treat, to transplant kidneys than to treat diseases. So yeah, like you, I'm very excited about this. I think it's going to be incredible.
And there are more than, I mean, just to put some numbers to it, in the US alone, I think there's more than 100,000 people at any one time on the national transplant waiting list, right? And people move to dialysis and outcomes become really, really poor if you spend too much time on the waiting list, right? So this is a really big problem to solve that's getting bigger every year, probably, right?
Yeah, and I'm very excited about an innovative new approach. Like I know a lot of your listeners are just such innovative people, and I think that's what we need in medicine. These big steps forward like this are just incredible, and our patients, I can tell you, they're really keying on this, and they're super excited.
Yeah, I mean, I certainly am as a as a person I've mentioned before who carries the ApoE4 who may be on the kidney transplant track in 10 to 20 years. I would love to not have to go begging and borrowing from family and friends to get one and have an easier path. So that's I'm rooting for the science on this one.
Yeah, I'd just like to say, I mean, I think there are several things like pig kidney. I think immunosuppression-free transplantation. I think that'll be something great. And I think medical therapies for these diseases. And then hopefully at some point, CRISPR. And so I think one thing like I always tell these families that I work with, the disease that you have, that you've seen in your parents or your grandparents, that's not the disease you're going to
Mm.
experience. Several generations ago, polycystic kidney disease really couldn't be treated. Then we had dialysis, but it was tough. Then we had transplant, but we couldn't help with rejection. Now transplants are even better. So with every generation of patients, things are better. And so for young people with kidney disease, I think the future is super bright.
Yeah, amazing. Thanks in large parts of the work that you both and your teams are
Yep.
doing.
Thank you.
You made a call out already for people to get in touch if they know anything about how to block production of these kind of proteins in the intestines or have other creative ideas. Is there and obviously kidney genetics collaborators you should be sure to reach out. Is there anything else that you want to leave people with, Stan or Tony, before we close out?
I would just like to say first, and then Stan, we'll let him finish off, but I would like to just say, I think we're going to be having this published around Rare Disease Day, which is February 29th, the rarest day of the year, of course, but it'll be 28th this year.
That's right.
And I'd just like to personally thank everybody out there who does research in these rare diseases. And I know it's difficult and it's not always fruitful, but you really keep our patients inspired. And, thank you for all you do.
Hard to follow that, Stan. Anything to add to that great message?
Yes, I agree.
Yeah, great. I agree too. As someone who's benefited from this research, and you get all kinds of unintended consequences, right? Besides helping the families directly, I know that your colleague with the ApoA4 license plate probably learned something fundamental about ApoA4 biology as part of this work, right? Because the rare disease can be a lens into some of these more basic science truths. So I think it's a great message to leave everybody with. And thank you both for your time today and all the great work that you do.
Thank you very much.
That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selective Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.